Okay, so last but not least, it's our pleasure to meet as our last speaker of today's sessions. And she will talk about characterization of bacteria isolates from antibiotic treated soils. It's a different story, so we have a whole variety of different topics we're going to cover today. And it's my greatest pleasure to give the stage to you. Please go ahead.
Thank you. Hi. My name is Adelike and you're welcome to my talk. So I'll be talking about the characterization of bacteria isolates from antibiotic treated soils. Before I go deep into what I did, I want to give you an overview of the importance of soils. Soils are important to us. We all know that. They support all living creatures. They are storage sites of primary substances and waste, and they are also sites of industrial as well as agricultural productions. Soils are sites of organic matter transformations. Soils are home to numerous microorganisms. For example, we have protozoans, we have fungi, we have a lot of them. We have algae, but we also have bacteria. But we are interested in the bacteria members in soils because they occur in various forms. Some are round, some are filamentous, some don't have shapes at all. Some are like rods, we call them rod shapes. So the majority of them are motile, while some are not motile. The bacteria communities in soil perform various functions, ranging from the recycling of nutrients to improvement of soil fertilizers. They help to form humus, they degrade recalcitrant stubborn substances in soils, and they also help to carry out biogeochemical cycles and so on. I would like to talk to you a little bit about antibiotics. Antibiotics are substances that inhibit bacterial growth. They have various mechanisms of functions. Sometimes they inhibit protein synthesis, sometimes they inhibit nucleic acid synthesis, which is DNA or RNA, and some of them destroy the cell wall. This is our normal bacteria. So some of them destruct the cell walls of the bacteria, and once the cell wall or the cell membrane is disrupted, then the bacterium is dead. So we are majorly interested in the effects of antibiotics on the bacterial community compositions in soils. So we did a column experiment whereby we collected soil samples from an agricultural area in Oldenburg, and then we carried out a column experiment by filling the soils. In the columns here, our experiments were done in triplicates. That's why you're having 3 of these. So we amended a part of groundwater samples that were collected in the same sample region. So we amended a part with 100 microgram per liter of incomycin. We also amended the part with 100 microgram per liter of sulfadimidine, and then we passed them through the columns and we collected the outflowing water, and we did so many experiments with the outflowing water. So we first of all extracted the DNA to see the community composition, and then we sent them for sequencing. We also cultivated some of this in nutrient agar containing higher concentrations of the 2 antibiotics. So what did we find out in our background studies? We saw that the inclusion of antibiotics affected the bacterial communities. As you can see that in the original soils, we have a lot of bacterial communities here, ranging from Acidobacteria to Actinobacteria. And then when you look at the soil samples with lincomycin or sulfadimidine, you can see that this community changed. We have some communities were excluded, while we have some communities that became so abundant. On our nutrients agar plate, you can see that we only had 4 communities, which was really expected. Of major interest were these Rhodanobacter species and Atrobacter species that we found to be highly abundant in sulfadimidine treated samples as well as in lincomycin treated samples, while they were not so abundant in our blank samples. So we got curious about the two bacterial isolates, and then we wanted to know what could be responsible for their abundance in the presence of high concentrations of antibiotics. So we estimated that the two isolates could utilize acetylnitrile as a sole carbon source. So let me just say tell you a little bit things about acetylnitrile. Acetylnitrile is an organic solvent that was used to dissolve our antibiotics because the concentrations that we used were not really soluble in water, and then we had to use this compound here. And because it is an organic compound, it contains carbon atoms, and we thought maybe the bacteria were using this carbon atom as a sole carbon source. And then we also estimated that the two isolates, the two bacteria, will be susceptible to higher concentrations of lincomycin and sulfadimidine. So we carried out a carbon preference test, whereby we subjected the two isolates in either glucose or either acetylnitrile, and then we did this test in two different environments. At first, we put them in the oxygen environment, and then then we later put them in the anoxic environment, which is without oxygen. So in the oxygen environment, they have oxygen, so they could breathe. In the anoxic environment, they don't have oxygen. Then we had to give them nitrate so that they would be able to breathe. And what did we find out? We saw that under the oxic environment, our Rhodanobacter species and Atrobacter species could really grow very well, but only in the presence of glucose. They could only utilize glucose as a sole carbon source, which means that they couldn't utilize acetylnitrile. So we have on our y-axis the OD measurement, which is the optical density that measures the growth of a particular isolate in broth. And then this is just the numbers of days that the measurements were taken. And under the anoxic environment, we saw that Rhodanobacter couldn't really grow. It couldn't even grow at all, both in glucose and in acetylnitrile. And Atrobacter, the same thing, but Atrobacter species was growing at first, and then it later died. So this means that Rhodanobacter species is an obligate aerobic organism, which means that it can only grow in the presence of oxygen, and Atrobacter species is a facultative anaerobe, which means that it can grow very well in the presence of oxygen, but it can only strive to grow in the absence of oxygen. So we concluded that both isolates prefer glucose under aerobic condition, but none of them could degrade acetylnitrile. So our study, this particular one, negates our hypothesis that the high abundance of Rhodanobacter species was as a result of their ability to utilize acetylnitrile as a sole carbon source. And then next, we want to see if the two bacterial isolates will be susceptible to high concentrations of the two antibiotics. For example, we had a very high concentration, which is 100 milligram per liter. That is so high. And then we also have 10 milligram per liter. So we subject this 0 microgram per liter was just our control. So we cultivated the two of them in different concentration of the two antibiotics. And what did we find out? We saw that the Rhodanobacter species was really striving to grow in the presence of high concentration of lincomycin. You can see here that this is still the OD, the y-axis is our OD, and then the x-axis is the numbers of days that the measurements were taken. We saw that here we in the 100 milligram per liter, the cells were growing. I mean, they were supposed to like go down like this. They were supposed to be dead, not to be growing. But we also saw that they also grew very well in the values of 100 microgram per liter. The same thing can be said about Atrobacter species. We also saw that, yeah, the same trends. We saw this is the blue line is the control. You can see that the sorry about that. The amended ones were really growing as our controls. And also in sulfadimidine, we saw the same trend. We saw that Rhodonobacter species was growing at a point and then it declined, but it didn't die. And the same thing was also seen in Atrobacter species in sulfadimidine in 100 microgram per liter. It was growing and then the growth was altered at first, but on a normal day we were supposed to see something like this. And so we concluded that the resistant patterns of Rhodonobacter species and Atrobacter species in higher concentration of liquamycin were really observed, but the growth was halted, but the organisms didn't die. But the mechanisms of resistance is quite unknown. Thank you.
Okay, you're well in time. You still have 3.5 minutes left. Okay, then we can start the discussion.
So honestly, I had some problems to follow your talk. So I did not understand why exactly you did these experiments. So why is it important to know that? And why you are focused on Rhodonobacter and Atrobacter and why these two are so interesting.
Okay, thank you. Normally, the bacteria, we have a community of bacteria in the soil. They're supposed to be diverse. They perform various functions in the soil. So the inclusion of antibiotics in the community could do lots of damage to the community compositions of the bacteria members in the soil. For example, antibiotics could remove some important communities and it could allow the overpopulation of certain community. As you can see here, that in the original community, we have lots of, in the original soils, we have lots of community compositions here, but when they were treated, you can see that this is actually protobacteria became so abundant while some communities were faded out, like the antibiotic removed some communities while some became so abundant, and that is not good for the soil ecosystem. So the reason why we focused on this two was that they were really, really abundant in our sulfadimidine as well as in our lincomycin soil, treated soil samples. Instead of they were not supposed to be there. I hope I'm able to answer your question.
Maybe I have to ask another question later, but I have to think about this.
Okay.
So actually, very nice talk. I was able to follow almost all of your talk because I have a background of microbiology, but I think it was difficult for the audience who don't have the microbiology background to follow that. It's true. Actually, the same questions that Sabrina want to ask, maybe I can rephrase. I know that there are some bacteria in the soil that do nitrogen fixations,
Yes.
and this is very important for the plants to grow. But what are these Rhodonobacter and Arthobacter doing in the soil?
Okay, thank you. For example, these two, we don't know what they are doing in the soil, but we know that Rhodonobacter species help to degrade complex organic substances in an environment. For example, if we have like long chain hydrocarbons that are not good for the environment, these organisms help to decompose them. And then, like, do you know something about bioremediation? I think I mentioned some of the functions in the second slide, I guess. Yeah, that they help to degrade. They degrade stubborn or recalcitrant substances in the environment. So that is one of their functions. And also, the Atrobacter species majorly is involved in nitrogen cycle, like you said. So the abundance is also very important in the soil.
All right. So the second question was, I think later I find out that what is that I think in your I forgot to mention the slide, in your previous slide I think you used an abbreviation LIM and SDM that I later found out that here.
Yeah, that is sulfadimidine.
Yes,
Yeah.
a little later I found out that at this moment I was not clear what this abbreviation means. And secondly, my question is that these antibiotics that we use, these vancomycin and the I forgot the name of
Sulfadimidine.
SPM. Yes. So what is the mechanism of these antibiotics? Either they're targeting the cell wall, the nucleic acid, the cell membrane, how these antibiotics actually work?
Yeah. Okay. Thank you. Sulfadimidine is known to inhibit the production of folic acid by certain microorganisms. That is one of the mechanisms. That is the reason why the majority of them are really, the two of them were resistant to sulfadimidine, because folic acid is being produced majorly by gram-positive bacteria, but we were working on gram-negative bacteria. So that is one of the mechanisms that sulfadimidine, one of the effects of sulfadimidine on bacteria. But for incomycin basically destroyed the cell walls of both gram-negative and gram-positive bacteria.
Okay, so still all of both of these.
Mm-hmm.
Okay.
Yeah.
And this was my second question, that how would you then find out from a soil that which is what bacteria? Are you doing some kind of sequencing of this bacteria or some kind of testing?
Okay, so what we do is to get the environmental sample, and then we just extract the DNA, and then we send it for sequencing. So that is how we know this is who is there, and this is who is doing what in the soil.
Alright, so from the soil you isolate a kind of uniform. You will get a DNA from several different species of microorganisms in that soil samples, and that is a kind of mixture of DNA from.
We have mixtures of DNA. We call it environmental genome sequencing or a metagenome. So we sequence all the communities, and then we can, from the sequencing result that we have, we can differentiate this is this and this is that.
Okay, nice, very cool.
Yeah.
And my last question is your graphs at the end. Yes, here, maybe, yes. So here we can see that the OD for your Orthobacter bacteria, your OD is something between 0 and 0.3. But if you go further into your slides,
The up or down?
yes,
Yeah.
maybe more.
I think maybe for the antibiotics.
Yes, here. now your OD is actually is coming between 0.1 to 0.25, even for your control.
Yeah. Normally, Rhodonobacter species is has a very slow growth rate. It doesn't really grow. It takes ages to grow. So that is why we're having a very low OD, and then this is in the presence of antibiotics. So we do not expect to have higher OD because we expect that antibiotics should be inhibiting the growth. That is why we are having lower ODs.
So the higher the OD, the more the growth, right?
The higher the growth, the higher the OD.
Yeah, the higher the growth, the higher the OD. And that's why I'm asking. So in your previous slide where I asked questions, OD for the control is also very low, right?
Okay, yeah.
And now that with the slide that we are discussing, the OD for the control is also raised up.
Yeah, so sorry I didn't mention that. In the control, we didn't have any glucose or any carbon source, and it's like we were not feeding them. So when you are cultivating bacteria in the lab, you're supposed to give them food, and that is why we had glucose. That's why you can see that the OD was quite high, but in the control there was no food. There was neither glucose nor acetylnitrate. So they were starving. We didn't expect any growth in the control.
Okay. Thank you.
Yeah, you're welcome.
So maybe it's a stupid question, but you showed that different types of antibiotics have an effect on the composition, the microbiotic composition of the soil, right?
Yeah.
And why is that important to know? So is it that you show that if you put too much antibiotic on the soil, the diversity is decreasing, or is it that you show that if you put the one or the other antibiotic on top of it, that you maybe can have a positive effect, that for example specific metabolic byproducts will increase, so that maybe that could be a benefit?
Thank you for that question. That is a great question. So we were interested, like I said before, that we got our samples from an agricultural area, that is where they rear animals. And you know that rearing of animals, everywhere they use antibiotics to feed animals, either to promote the growth of these animals or to prevent prevent them from getting infected with specific bacteria. So when these animals are being fed with antibiotics every time, they excrete these antibiotics in the environment, in the soils, and this gets washed into the marine environment. But we wanted to know that, okay, the inclusion of these antibiotics can affect everyone in general, not only the soil, but it can affect us as humans, and it can affect the organisms in water. So that is why the studies is very important, that we show that if you continue feeding animals, or maybe fish or any other organism with antibiotics, then you are likely promoting antibiotic resistance among animals, among humans, and also you are affecting the community compositions of the bacteria in the environment.
I have a question to this figure. So what is actually on the y-axis?
On the y-axis, we have our optical density.
Optical density.
Yes.
Okay. And then you show estimates and some kind of standard error or standard deviation, or like this arrow, the black arrow bars. What are these?
Yeah, they are standard deviations. We took the measurement in triplicates, and then we calculated the standard deviations between the triplicates.
occasionally. Although here, especially to the right, it's rather obvious that the proposed treatment is different from the other ones. Have you conducted any statistical tests to?
No, sorry.
No, you haven't.
No, yeah.
Okay, one more question. Are these data from single biological N?
Single what?
Is it data from the single biological N? I mean, how many times have you repeated this experiment independently?
Well, it was my master thesis, so I did it once. I only measured once.
But when you have an arrow bar,
Mm-hmm.
this is based on a certain number of measurements. This is what you refer to, right?
Yes, but these are the technical N's, means you are conducting an experiment at the same time,
Mm-hmm.
but in triplicates.
Yes, I said it,
Yeah.
like in triplicate, so we
But
have
But what if there is a general problem happens and the effect that you are seeing is not the real effect, but an artifact? And well, our lab is saying that one experiment, one N is worse than no N. Because you have to reproduce your data at least 2 times. So this data is still one biological N. Even you have 3 triplicate, technical replicate, right?
Okay. Yeah, we're really paying attention to the deviations, really, because like I said before, that it was my master thesis, and I measured in like triplicate, and I only took the deviations between the. So I think we were not really paying attention to the statistical differences.
This is also problematic for calculating the error, right?
Yeah.
It's based on just sample size and 3 replicates.
Isn't it? Because when we do some experiments, as I have to do some experiments, and each experiment I have to go for triplicate. So, and then I have to calculate the mean from all those triplicate, and then I have to repeat this experiment 2 times. In every time in triplicate, and then I will be able to find the standard deviation. And that would be a reliable readout, isn't it?
Further questions?
Maybe to this one here actually. So if you have the triplicates, what they're called, and if you take the mean and then doing the data analysis, I mean you could in principle also just put everything together. And so it's basically a question about the signal to noise ratio. If you want to do the average first of the small sample size, then that might be an advantage. So, but if your signal to noise ratio is actually good.
To be honest, I couldn't understand.
So the main thing is, are you doing tests in small sample size of 3, repeating that over and over again, and then take the mean of the small group of the 3 samples, or you could just put everything in one big vector, for example, and ignore the 3 split. And sometimes we have you have a big better estimate for, so you basically increase the N if you put everything together. But the disadvantage is if you have a lot of noise in your data, then you might be better off taking the small sample size. And that's the main question, how to decide which one to choose.
Well, if you don't know the signal to noise ratio, then you should repeat it, right? And don't put it together.
You still have to analyze it, but you can do everything at once. So you don't need to split this 3 segment, I guess.
Yeah, well, different field. In my field, we would actually prefer to have many samples and then estimate the mean and then the variation of our estimate, instead of putting all the stuff together and calculating based on that.
Yes, because you have extremely high noise.
Sorry?
Because you have extremely high noise in behavior data.
Yeah, but if you don't know the signal to noise ratio, I can't say this for this time because I haven't got a clue about that. So I haven't got a clue about the signal to noise ratio. But I would say if you don't know that, I guess I would follow my strategy. I mean, with Corona, they also
I just want to point out that it might be an alternative, and sometimes it's advantageous, and so it depends on your data which one is better. So if everyone in this field uses this, there's no choice anyway, so you have to do what everybody's doing.
Yeah. Actually, for example, if you can go to one of your graph,
Okay. Which one?
yeah, for example here, in right panel of your figure for the orthobacteria where we have seen a very nice curve for the glucose, right?
Mm-hmm.
And what if there is something wrong with your stock preparations or your working dilution preparations? You have used more glucose or maybe you contaminated your solution with something else and you don't know. And then the same solutions you used in three triplicates, and that's obviously giving some or more or less the similar results. And then you can, your data is saying that, okay, then you add this glucose, maybe it's a real effect, maybe it's something coming from contamination, and you got a spike, a very nice growth. And to eliminate this point that, okay, this is not because of something else, but purely to the glucose. It would be nice, in my lab, what we do is to repeat the same experiment 3 different times. And when then the data is coming, fitting nicely, we can rely that, okay, yeah, this effect is because of the glucose, not is something from a contamination or something else.
Yeah, I was supervised, so I really did it according to how I was supervised.
Do you have a question? Okay, then we go again for the competence stuff, and if you could improve for the next time.